This is the second video in a series on how to write scientific papers. In the last video, if you remember, we discussed how to write an introduction for a scientific paper, and in this video we're going to discuss how to write an abstract. So we're going to go ahead and follow the same procedure as we did in the last video. That is, we'll go ahead and talk about what it is we want to include in our abstract, and then we'll look at an example abstract that has been published in a scientific paper that we will then use as a template to build an outline. So let's go ahead and start talking about how it is we want to write an abstract. So because online research databases typically contain only abstracts, it's vital that you include a complete and concise description of your work in your abstract so that you can, 1, entice potential readers into obtaining a copy of your full paper, and to also make it simple for readers to quickly decide whether or not your research is going to be helpful to them. You want each sentence to say something important. Don't include any unnecessary information. After all, if your paper were to get published, most of the time the only part of your paper that is going to be read is your title and your abstract. Hence why it's so important to get good at writing abstracts. So if you remember from the last video, when we discussed writing introductions, we looked at this paper titled Directed Evolution of E. coli Alkaline Phosphatase Towards Higher Catalytic Activity. So we're going to go ahead and continue using the same paper as our source for writing the outline for our abstract. The way that we're going to do this is we're going to analyze each sentence and then figure out what purpose that sentence is serving in this abstract. So let's start reading. Directed evolution was used to enhance the catalytic activity of E. coli alkaline phosphatase. Alright, so in this first sentence, it seems that the authors are simply identifying the problem that it is that they're trying to solve. So this is a really good way to open up an abstract because not only do they tell the reader what problem they're addressing, but they also let us know what the scope of their work is as well. Now, over here on our outline, we're going to construct this in such a way so that it will be as though we're going to answer a series of questions that will help us write out our abstract. So, based off of our example, let's go ahead and write out the first question we want to answer, which is, what problem are you trying to solve? So this is the first question that we're going to try and answer when we go to write our own abstract. Now let's go back over to our paper and continue reading from this abstract and try and figure out which questions these authors are trying to answer next. Through 2 rounds of error-prone PCR and 1 round of DNA shuffling, followed by a rapid sensitive screening procedure, several improved variants were obtained. After reading this second sentence highlighted in green, we find out in a very brief fashion how they went about solving this problem. And in addition to that, we also get a brief overview of the methods that they used. Error-prone PCR and 1 round of DNA shuffling, followed by a rapid sensitive screening procedure. Also, note how they say several improved variants were obtained. So already we know by the second sentence that their experiments proved to be successful. So if we're following our example, we'll go ahead and write the second question we want to answer as, how did you go about making progress on the problem? Alright, so when we're writing our abstract, the first question we're going to try and answer is what problem are you trying to solve, and the second question that we're going to answer is how did you go about making progress on the problem. And once again, this is done very briefly in 1 sentence, the way it is done in the example abstract that we've been reading from. Now let's see what the third sentence tells us. Their enzymatic kinetic properties, thermal stabilities, and possible mechanism for the improvement were investigated. Alright, so we didn't really learn anything new from this third sentence, but it does seem to be a continuation of the second sentence. That is just more information on how they went about making progress on the problem. So let's go ahead and highlight this in green in order to show that. So let's go to the fourth sentence. In 1.0 molar tris buffer, the specific activity of the most active E. coli alkaline phosphatase variant, S2163, was 1500 units per milligram protein, showing it to be 3.6 times more active than the D101S parent enzyme, and approximately 40 times more active than the wild-type E. coli alkaline phosphatase. So now we're starting to learn a bit about the results. Something important to keep in mind when you go to reveal your results in your abstract is that you don't want to do it in such a way that it's going to be misleading. So notice how they don't simply write the catalytic activity of E. coli alkaline phosphatase was improved with this mutation. Instead, what they do is they report the specific numbers and compare the activity of each of these enzyme mutations to all the rest of the mutations and to the wild type. That way there isn't going to be any discrepancy about what these results are indicating. So these next few sentences appear to go on listing the results, so we'll continue reading. At the same time, the Km value of the S2163 variant decreased to 1491 micromolar from the 2384 micromolar of the D101S. As a result, the Kcat over Km ratio of this variant showed a 5.8-fold enhancement over the D101S parent enzyme. So I'll go ahead and highlight these next few sentences to show that they're part of this results section. And once again, notice how they're explaining the results in the context of the experiment and they're using specific values. So back on our outline, let's go ahead and write our next question to be answered as what's the answer? And let's go ahead and make a note that reminds us to provide results that cannot be misinterpreted. And also remind ourselves to be specific when we're reporting values. So before we go on and read these last few sentences, I want to point out that it's also a very good idea to explain what your results mean when you're writing an abstract. So as you can see from this example, the best way to go about doing that is to report your results in the context of what they mean. For example, when the authors say that the Kcat over Km ratio of this variant showed a 5.8-fold enhancement over that of D101S parent enzyme, they aren't reporting the actual value that they got for the Kcat over Km ratio, which may not make sense to scientists outside the realm of biochem, so instead they just cut to the chase and tell us what that value means, which is a 5.8-fold enhancement over the D101S parent enzyme. So with that in mind, let's go back to our outline and just write our next question as, what do your results mean? Now, let's go ahead and finish up this abstract. 3 activating amino acid substitutions, K167R, G180S, and S374C, which were located far away from the center of the catalytic pocket, were identified by sequencing the genes encoding evolved enzymes. Possible explanations for the improvement of activity were analyzed. Alright, so it looks like these last few sentences are just more explanation on how they went about evolving E. coli alkaline phosphatase. So we'll just go ahead and highlight these parts in green. Now let's go ahead and take one last look at our outline for how to write an abstract. First thing you want to do is answer the question, what problem are you trying to solve? Then answer the question, how did you go about making progress on the problem? Next, report your results, but keep in mind that you want to provide results that cannot be misinterpreted and also be specific with your values. Finally, be sure to tell your reader what your results mean. Alright, that sums up this video on how to write abstracts for scientific papers. I hope that this outline will come in handy when you're writing your own abstracts, and thanks for watching.